So I think we finished off I was giving you some examples of some successfully used and marketed and sold genetically modified products. So this had included these Arctic apples here, which have been modified so that they don't brown the way normal apples do. This virus-resistant papaya, which apparently has saved the entire papaya industry in some areas. So this was a huge, huge success, and papayas are pretty lucrative crops, I hear, as well. And then the third one I showed you, which I had read about this being in Canada, it's definitely in Quebec. I don't know whether it's across Canada or not, is the genetically modified salmon. So let's talk about a few other genetically modified things, because I know at this point, you know, people usually have different kind of emotions around genetically modified foods. You know, some people are like, okay, never ever, you know, I don't want to touch those Franken foods. Other people are like, wow, this is really cool. You know, there's a lot of different opinions around genetically modified foods. There's certainly a lot of potential there. And if we can make fish a lot bigger, maybe we can feed more people on the planet. But let's just talk about genetic modifications. I thought I'd show you also these genetically modified strawberries here. And if you're familiar with strawberries, then maybe you realize that what we're actually looking at is just domestic strawberries on the top. So these big ones here, and these other ones here are just wild strawberries. So why am I calling these genetically modified? Because the domestic strawberries have been modified. Humans have taken these, we've domesticated them. We've, over many generations, we've grown these things, selectively bred them. And if you look at the genomes of these things, the wild varieties, their DNA and their genetic sequences is actually different from the tame varieties. So part of the argument for using labs to genetically modify things, of course, is that, hey, we've been doing this for a long time. What's the big deal, right? Let me show you a couple of other interesting examples, because strawberries, it's pretty easy to see that a strawberry is a strawberry. Maybe they're bigger. You know, that's not a big deal, right? Let's talk about corn. So for a long time in modern history, it was a mystery as to where corn actually came from. I think it was finally proposed in the 1960s that this plant here, so teosinte, I think, teosint, I'm not exactly sure how to say that, was a wild variety of corn. And if you look at the genomes, I think there's even a different number of chromosomes here. And they're nothing like it. Like modern corn, if you know anything about corn planting, this crop cannot actually survive in the wild. It actually requires humans to propagate it. There's many, many varieties. They make massive kernels. It's the #1 food crop in the United States and several parts of the world. And it's soft and, like I said, many varieties. This teosinte is like a grass, and the seeds are tiny and they're hard. If you want to eat it, you have to grind them, and they're very, very small. And so, like I said, the mystery is how on earth did humans ever take this crop and change it so much? It must have taken 1,000 years. I'm not sure. But if you like corn, we're grateful for this, but the amount of genetic modifications are massive. And so not, you know, way, way more different than some of the minor tweaks we're doing in the lab nowadays, in many ways. Here's another crop that you may be familiar with. You're probably looking at that and thinking, no way. But this actual species here, so Brassica oleracea, I guess, actually makes a whole bunch of different modern food crops. This is all artificial selection. Select for the flower, select for the leaves, select for different colors. We'll get a whole variety of different vegetables. They're actually all the same species. So this is really, really incredible when you think about it. And like I said, the argument is that genetic modification that we're doing nowadays, we're just accelerating the process, and we're actually not making as many drastic changes as have been done in the past to some of these crops. So just some things to think about. I just wanted to finish off this topic and show you 3 exciting CRISPR-Cas9 projects. I'd mentioned that CRISPR-Cas9 is is really an incredible technology because we can actually modify DNA in cells. And most of these modifications, they're not actually transgenic. We're not necessarily introducing new genes. We're just modifying existing ones. So, some people are against transgenic organisms because, you're taking an organism from, let's say, a jellyfish, and you're putting it into some sort of zebrafish, and that's two different organisms. And there's something that's kind of unnatural about that. In this case here, we're making tweaks. So you can see here's an example making hornless cattle. People are probably thinking, oh, what's the big deal? Well, talk to farmers who deal with cows. They have to dehorn them. It's a tough process. Some cows can bleed to death. It's dangerous. And if you don't do it, they gore themselves. And so why not just make a hornless variety? And actually that was done just a couple of years ago. What about combating infectious disease? We're talking about infectious disease all the time right now. And, one of the scourges of the world, thankfully not in Canada, is malaria. And malaria is a bit of a tropical disease. So one good reason to live in Canada, that's how we get rid of our tropical diseases, is we have winter. But malaria is a big deal. And many, many people get sick annually, and millions of children die from it. So why not just, modify the mosquito? And so in fact, there's a couple of projects working on this to make the mosquito so it can't carry the parasite. I don't know a lot of details about this, but I know it's a CRISPR-Cas9 project. What about modifying humans, right? This is, another area where, you might start to think about the ethics and questioning whether you should do this or not. But there are some people that have diseases like HIV, and so it's a lifelong infection. And so the thought is that we know that we could do this. We could take somebody's immune cells and we could make them resistant to HIV. So you could give them a whole immune system transplant, and in theory this could be done. And you can actually read about it there in that article if you want to see what has been done. Hasn't been done in humans that I know of yet, but like I said, it's sort of at a hypothetical time. And if you have HIV, this could be your thing in the future, and that's kind of exciting. Okay, so I had mentioned on the final exam, you're going to see one of these questions. It'll probably be a multiple choice question, looking at these forensic gels, and so it'll be a multiple choice question. So it's going to be phrased like, which children belong to the father, and A will be, child's 2 and 3, B will say child 1, 2, and 3, and so on. And you have to read, the question carefully to figure out what is going on. This one here is labeled really well, and it says which child is not likely to be the offspring of the father. So always just double check, right? Just to see that the mom has, half of her band should match with each child. So I'm just checking that here. And child 3 over here for child 4 is matching there, and child 3 is down there. So let's take a look, and we got to look at dad and see if dad has half of his DNA matching with mom. So here it looks like child 3 and child 4. And what about child 2? So child 2, right? Nothing here and nothing here. So what about child 1? Let's just double check child 1, and child 1 is okay. So the answer in this case here would be child 2, because none of the DNA bands match. So hopefully you find that relatively straightforward. That's one question you're going to see on the final. And just make sure you do read it carefully. It might not make it quite as easy as this. So before we talk about viruses, I just want to kind of talk about the final exam for a minute. So this is kind of the layout of the final exam, or at least the planned layout, is that I'm going to have 80 marks. So it's a little bit more than double your midterms, but in terms of length, it's exactly double, so 2 hours. There's not quite as much written as 2 midterms. So 40 of those marks will be true and false or multiple choice. And so the way I do that is we have 20 topics. So basically I go and I take about 2 questions a topic, so through the entire course. And most of the rest of the questions are going to be from the last part of the course. So starting at topic 13. So topic 13 was when we started talking about DNA and structure of DNA, and then everything that we've covered since, and including the virus topic that we're going to start today. So all the written and everything, there may be some questions that are kind of a little bit more, they might cover some things that we've talked about throughout some of the units throughout the entire course. I think I mentioned a couple of times that one of the themes of this course is discussing differences between prokaryotes and eukaryotes. So, you know, that's something that kind of covers the whole course. And, you know, if I think of something else along that range, I might come up with a long answer question around it. So it's going to be worth your time to go through topics 1 to 12, but don't spend the bulk of your time there. Spend most of your time studying topics 13 to 20. And I do encourage you to go back through the lectures and see what I've said. I've given a lot of hints about the final exam. Okay, so enough about the final exam for now. What I want to do is talk about viruses today. And like I said, this is one of my favorite things to talk about. So just going to get that PowerPoint up. Hold on a second. Almost there. Where's the share button? There we go. Okay, hopefully you can see that now. So we're going to talk about viruses. And like I said, I love talking about viruses. I think they're super, super fascinating. And I wish we had more time in this course to cover them. But you're going to get about a lecture and a half on viruses. And so usually when I cover this unit, I talk about a whole bunch of things. I talk about viruses for the most part, and then I talk about HIV and influenza as kind of case studies because they're interesting viruses. And then we talk about a few other things that sort of fall under this category of self-replicating entities. So I talk about something called viroids, transposons, and prions. I'm not going to cover these things this semester, okay? I'm not going to talk about prions or viroids or transposons. You can read about them in the textbook. Instead, what we're going to do, and like I said, that's going to be covered on Monday, is we're going to talk about COVID-19 and coronaviruses, and we're going to talk about vaccines. So I'm going to kind of substitute that. I think it's just a little bit more important right now that we talk about what is going on in the world. And, you know, as science students and students taking biology courses, it's part of your job to combat misinformation. It's part of your job to, you know, help people understand what is going on. There's a lot of misinformation out there, which is really unfortunate. So we'll get to that Monday. Like I said, COVID-19. Today, we're going to kind of generally talk about viruses and talk about what they are and what they do. So I usually ask people, you know, in class, and, you know, I look everybody in the eye and I say, have you ever had one of these virus infections? And, you know, people probably assume, yeah, okay, probably something at some point. And then I ask them, well, what about this virus? And of course, nobody knows what that one is. And I tell them this is a herpes virus. So I ask the students if anyone has herpes. And of course, you know, everybody says nothing at this point. And then I turn around and say, well, I'm here to tell you today that all of you have had herpes, or at least a herpes virus infection. And so let me explain. So it turns out there are actually 8 herpes virus infections of humans. The first 2, you can see on this list here, we have herpes simplex 1 and herpes simplex 2. And these are known for causing cold sores and genital herpes. So generally, cold sores are usually caused by herpes simplex 1. And genital herpes is usually caused by herpes simplex 2. But both of them can actually cause both infections. And so over here on the right, this number here is the prevalence. So what is prevalence? It means that's how many people in the population we think are infected with these viruses. So maybe about 60% have cold sores, and some people have it and don't even know it because you don't always have the symptoms. Genital herpes, maybe 10 to 20% of the population. In Canada, I don't know how good the numbers are for in Canada. I think they're lower. I think they're like 8 to 10%. So good job, Canada. So what I'm telling you right now is there's a 60% chance you have one of these herpes viruses, maybe a little higher. You could have both, I suppose. All of these cause blisters that are unpleasant, and they're lifelong infections. So that's why I'm saying you're infected right now if you have one of these. If you don't have one of these, there's also a chance you might have one of these. So you can see both of these are 95% prevalence. Both of these, the numbers are changing. You can see the first one there, varicella zoster virus causes chickenpox. This used to be a rite of passage when you were a child. Everybody got chickenpox, usually, you know, around age 5 when you started to go to school and all that. Now, all of you are probably young enough that you've been vaccinated and you probably don't have it. So, you know, that number, I'll have to update at some point, but I don't know if we actually have any good studies on that at the moment. Epstein-Barr virus causes infectious mononucleosis. By the time you are 30, you've got a 95% chance of having had it, and sometimes you don't have symptoms. So at this point, there's a really strong case that you are infected with at least 1, possibly 2, maybe even 4 herpes viruses. So you can bring that home today and tell everyone, you know, all this exciting news. So if you haven't had any of those, there's a very good chance you've had some of these herpes viruses here. Roseola, sometimes called baby measles, super common. Most people get it by the time they're like 2 years old. You can see this 6th disease, or actually this one here is baby measles, but they both end up with similar symptoms. Very good chance you've had them. And like I said, these are very, very common infections. Most of these viruses here, there's no symptoms or very mild symptoms, and you don't even know you had it. One more herpes virus actually infects humans. I'm assuming none of you have had herpes B because it's fatal, 100% fatal. So probably none of you have had that. That's actually from monkeys. So I find this stuff really interesting. We've got these viruses, they can infect people, and these are actually lifelong infections. And we'll talk about how that works. Hopefully we'll get to that today. So a few more virus facts for you, and then we'll get into some other details. We are exposed to billions of viruses daily. And you know, they're found in the air like bacteria. They're found on surfaces. If you have eaten a salad or some vegetables today, there's a good chance that you have consumed viruses that were there, and they got there because there were insects, and there were insects on those plants when they were growing out in the field. And there were viruses that came with those insects. So they seem to infect everything. Everywhere we look, we seem to find them. It seems that every organism that is out there has at least one virus that infects it. So there are plant viruses, chimpanzee viruses, dog viruses, cat viruses, you name it, platypus viruses, everywhere. There are viruses that infect E. coli, and we're going to talk about that today. And they're infecting E. coli and other bacteria in your gut right now. So I guess sort of the bottom line I'm trying to make here is that most viruses have little effect on us and little impact on our health. Many of them are infecting other things. We have great immune systems that protect us against a lot of these things, and that's something important to know. So back to this picture, I showed you this one way back at the beginning of the semester, and we were talking about the different kinds of cells. We've got a prokaryote up there at the top, and we have a eukaryote here on the left. This animal cell has a nucleus. Remember, that's what U for eukaryote means. It means a true nucleus. And I threw a little virus on here just to show you the scale. So there's the virus there. Very, very, very tiny. You can't see viruses with a normal microscope. You need an electron microscope. I know I have a zoom in here. This is actually the influenza virus. And you can see it says here it has DNA or RNA. And this one has an envelope, which is kind of like a membrane. And we'll talk about what that is in a minute. So first, let's talk about how small these things are. Very, very tiny. So there's a tall human, 2 meters. That's pretty tall, but it just makes the math easier. So there's the size of a rhinovirus. So what's a rhinovirus? Let's talk about that for a moment. What's a rhinovirus? You're probably thinking a rhino is an animal, and it has a big thing on its nose. And actually that word rhino means nose. So this is a virus that infects your nose. This will maybe give you the common cold. And that's where the rhinovirus is. So it's about 200 nanometers. And we can do some math. And 2 meters divided by 200 nanometers. So it's kind of like the math we do when we do magnification. You got to convert the units so that they are the same. And that's about, so we're about 10 million times larger than a virus. What does that look like? So there's the head of a pin, right? That's about 2 millimeters. And here's something, a couple of things actually, that we can just barely make out with our eye. The human eye, the resolution is about 100 micrometers, give or take a little bit. So these are some things that are kind of just visible with our eye. We can go really small, and we can think about a red blood cell, which is 10 micrometers, and it just looks like a speck compared to that, and we can't see that with our eye. It's a little too small. So let's compare a virus to a red blood cell, and a red blood cell is 10 micrometers, and the rhinovirus is about 0.02 microns. I think I have the number wrong there. I'm thinking it's 200 microns. But hold on, well, let me just take a look back there on that previous slide. I have 200 nanometers. Yeah, it should be okay. Well, I have the number wrong, but you get the idea. It's super, super tiny, right? It's 0.2 microns. So what does that mean? It means about 500 million rhinoviruses will fit on that pin, and one sneeze, and you can infect thousands and thousands of people. And this is why we have these mask bylaws. We're trying to prevent these respiratory droplets from getting all over the place, because potentially people can be shedding virus and have millions of them in one droplet. So what is a virus? This is a definition I took from, I can't remember if it's our textbook or not, but you can see it says here it's a minuscule acellular infectious agent having one or several pieces of nucleic acid. It's kind of confusing in some ways. We've already talked about minuscule, and one other way to compare small is to look at the genome. Here's the human genome. This is actually the haploid genome. We actually have double that amount of DNA. We're looking around 20,000-ish genes. E. coli is about maybe 4,000 to 5,000 genes. And your typical virus is much, much smaller. The influenza virus has, I think it's 10 or 12 genes. So not very complex in some ways, very, very small. So that other part, they're infectious agents, right? So they infect cells, and what they do is they hijack the cell. And all these things that we've been talking about recently. We've been talking about the cell replication, transcription, and translation machinery. So the genetic instructions from the virus get in the cell, and those genetic instructions basically say to the cell, hey, make more virus. So they're going to use all the cell's machinery, and understanding how that machinery works is important if we want to understand viruses. So a couple other things about viruses, and then I want to show you some other examples. I mentioned viruses are intracellular parasites, meaning they have to live inside cells. Let's compare that to a bacteria, which the answer is no. We can give a bacteria food, and it can eat food, and it can metabolize that food, and that's how it works. A virus can't do that. It has to grow inside a cell. Now, there are some exceptions. There are a few parasitic bacteria like chlamydia that live that way, and in fact, historically, initially, we thought chlamydia was a virus because it's kind of living like one. Do viruses have plasma membranes? No, and I'll talk about what that means shortly, because I mentioned that the influenza virus kind of has something sort of like a membrane. A classic definition, and you still see this in textbooks, so that's why I have it here, is that viruses are filterable. So they used to be called filterable agents, meaning they were tiny. Kind of a dumb thing to say, but I guess it just means they're small. Has both DNA and RNA? Viruses? No. Viruses have DNA or RNA. And I'll show you some examples in a minute. Can viruses make ATP? No, of course not. They don't have cells. They don't do respiration. They don't do any of that stuff. Do viruses have ribosomes? No, they use the cell's ribosomes. Why would they have their own? They're completely parasitic. They completely rely on cells to do every single process. And are viruses sensitive to antibiotics? No. So we talked about antibiotics, and we had mentioned that antibiotics are exploiting cellular features in bacteria, right? And so viruses don't have cells, so they're not going to be sensitive to antibiotics. So if you have COVID-19, or if you have the flu, or a cold, do not take antibiotics. They're not going to help. So what does a virus look like? Okay, so a virus has a genome. The genome is DNA or RNA. So you just have one or the other. And actually, they could have a variety of types, meaning we can have actually single-stranded RNA, we can have double-stranded RNA, we can have single-stranded DNA. So viruses kind of have a little bit more diversity there in terms of how their genomes work. Then a virus is surrounded in a protein coat. So we've got some genetic instructions, and it's packaged up in a protein coat called a capsid. Sometimes you might see people talk about viruses and they call them nucleocapsid. So you can see it just means the same thing. Capsid that's surrounding nuclear material, which is DNA or RNA. So I don't know why we have to have so many fancy words, but sometimes that's the way it works. And sometimes viruses have what we call an envelope. It's not a membrane. It's an envelope. A membrane, if you remember, we talked about all those things membranes do. They transport things, and virus membranes don't do that. So we call it an envelope. And it's something that it would grab on its way out when it's budding away from an infected cell. So this actually comes from the host cell as well. And those virus membranes have glycoproteins, and these are viral proteins, and they're usually involved in the infection cycle. So sometimes, and you see in the news with COVID-19, we're talking about these, and they're sometimes called spike proteins because they kind of stick out, and under an electron micrograph, they kind of look kind of spiky. So we'll see some pictures of some viruses here in a moment. So there's some different viruses. These are cartoons, obviously, and I'll have some real pictures and real viruses here in a moment, but you can see they come in many shapes and sizes, right? And they have all those features, right? So they're all going to have some sort of genetic material, and that genetic material, I guess, is in the red, and the capsids are in the blue, and so they all have that. The genetic material can be DNA or RNA, and you can see examples of that both. Some of them, just this one here, has an envelope, and you can see these little glycoproteins. They kind of look like little spikes sticking off the viruses. So I'll show you some examples here of some viruses, and this is just to show you all the variety, and you'll probably recognize some of these viruses. There's Ebola up there on the top, and it's an RNA virus, and it's helical, so it's kind of like a big tube, and the tube is kind of big and flexible, and it actually kind of ties itself in a knot, which is very interesting. I'm not sure if that has any importance biologically, but it's kind of cool. Some viruses are polyhedrals, so you can see they're made out of these triangles, right? These triangles are just kind of the capsid proteins, and they snap together, and this is called an icosahedral shape. And you can have a bigger genome if you have more triangles and make a bigger kind of shape, right? So there's a mastodon virus. I don't know what that is, but an adenovirus right here up top. That's something that also causes the common cold. And poliovirus, of course, causes polio. So there's a lot of envelope viruses out there, particularly when we're looking at human viruses. There's HIV right there. And HIV is kind of interesting. You can see it has a capsid. It actually has a second capsid called a matrix. I don't know why we don't just call it capsid 2, but whatever. It's got the membrane, and then you can see GP120. That's a glycoprotein. The 120 stands for the size, so that's the molecular mass. That's 120,000 grams per mole for that protein. It's pretty large. Influenza virus is another example of an envelope virus, so right down there in the corner. And coronaviruses are also envelope viruses. So this is something important to know because, of course, an envelope makes them relatively sensitive to disinfectants. So hand sanitizer does work on coronaviruses, in case you're wondering. It does not work on non-envelope viruses. And then there's some viruses out there that are just a little bit extra complex and fancy. And so the bacteriophage, we had talked about bacteriophage a couple of times this semester. This infects E. coli. And if you take a look up top here, this part here is your basic virus structure. You've got some DNA and it's wrapped in a protein coat, the capsid. But it has that whole bottom part. And it's It's kind of, you know, I know the whole thing looks a little bit like a fancy lunar lander or something like that, but you can actually think of it as a sophisticated syringe. So that, that part is going to dock on E. coli, and it's going to inject its genome through that tail section. Not sure why it needs it. It might have something to do with getting through the peptidoglycan cell wall, but I'm not entirely sure. So there's some pictures of some different viruses. Like I said, they come in all shapes and sizes. You don't need to know all those viruses and all their shapes and sizes. I'm just showing you the variety because I think it's pretty cool. You can see some viruses are almost big enough, but not big enough to see with the human eye. Like this is a pox virus, like smallpox, and compared to streptococcus, quite a bit smaller. So how many viruses are out there? Many. This is the most amazing thing to think about viruses. Let me just show you one virus, and show you how many are out there. Let's talk about HIV. So there's about 34 million people on the planet with HIV, which is unfortunate. And if you think about how many virus particles are in these people, there's the number, 10 to the 16. So that's a 1 with 16 zeros. I can't count that high. That's not as big as Avogadro's number, but my understanding is 10 to the 16 is big enough that if I had that many green peas, it would cover the globe in like a centimeter green peas. So that is a massive, massive number. And think about all of the other viruses that are out there. The numbers are massive, and they're all over the planet. It's really incredible. Okay, so again, also going way back to the beginning of the course, we were talking about some of the definitions of life, right? So we had all these definitions, and we talked about, you know, a little bit about what they were, and you know, we concluded that cells are alive, they have order, they reproduce, they can adapt from generation to generation, they metabolize energy. Let's just talk about viruses, because this is kind of one of these things where most biologists would say that viruses are not alive. They are infectious particles. You know, maybe you might loosely use the word life form, but you know, not technically alive. But let's see. So, are they ordered? Yes. Yeah, they've got those capsids. They've got geometrical shapes. Of course, they've got order. Do they reproduce? Now we're getting to that gray zone. They don't really reproduce on their own, right? They require some sort of host cell to do all the dirty work for them. So, still kind of a gray zone. Do they grow? Not really, they don't grow bigger. Do they metabolize energy? No. Do they respond to the environment? No. Do they undergo homeostasis? No. Do they adapt? Yes. Yes, viruses do. We need a new flu shot every year because, well, the virus mutates, so it adapts. So this is why most scientists probably would say viruses aren't alive, but you can see they are part of the biosphere. They do affect living things, and some people would say that, yeah, you know, some people would argue this, but it's a little bit debatable, but just something kind of to bring back some things we were talking about way at the beginning of the course. So I'm going to show you some pictures of some bacteriophage. A lot of what we know about viruses were learned and are still learned in bacteriophage because they are super easy to study. And they're not dangerous to humans. They're infecting bacteria only. So in order to study like a coronavirus, you need a level 3 lab. Well, how many level 3 labs do you think there are around here? Not many. I think University of Alberta has 3. I'm not sure if there are any at University of Calgary. So that means all of Alberta has like maybe 3 level 3 labs to study coronaviruses. So we study something easier, something that doesn't make humans sick. And this is where bacteriophage come in. So you're going to see on these pictures here how a virus life cycle works. There's a cool picture. This is, I can't remember if this is from our version of the textbook, or I think maybe it's from a previous version. Amazing, amazing scanning electron micrograph showing E. coli being infected by a bacteriophage. Really, really cool. So I thought I had a video here. Maybe it's coming up. Hold on a second. Let me just take a peek. Yeah, sorry, okay. So the video is coming up in a couple of slides. So I'll get to there in a second. I guess I gotta re-share my screen. So there we go. So I want to talk about virus life cycles, and this is really important in the biology of viruses, and there's kind of three general strategies. At any one time somebody talks about a virus biology, this usually comes up because it really affects how the virus and what it does to the body, right? So three strategies. One is called the lytic cycle. And these examples you can see are all through bacteriophage, but there are human examples of all these viruses. So basically the first step is the virus recognizes something on the surface of a cell. So in the case of a bacteriophage, it's actually recognizing, I believe, lipids or carbohydrates, maybe it's glycolipids on the surface of E. coli. And that would be in these little tail proteins here, these feet or whatever you want to call them. At that point, so E. coli has a peptidoglycan cell wall, and so this virus actually is an enzyme that digests that cell wall, and then it injects its genome. So in this case, it's a DNA genome, and this virus is really nasty. You can see it is actually destroying the host DNA. That doesn't happen with all viruses, but it does with some, and it's fully taken over the cell, hijacking it entirely. At that point, now you've got this virus DNA, and the virus DNA has instructions, and the instructions say make more viruses. So how do we do that? Well, it's DNA. DNA is going to get recognized by RNA polymerase, and it's going to make RNA, and the RNA is going to be recognized by ribosomes, and the ribosomes are going to make viral proteins. So you can see that cell there is loaded with all these virus parts. They assemble. Apparently, they kind of self-assemble. I don't really know much about that, but you know, you've got complementary intermolecular forces that are helping those things to kind of assemble together. We do know it's a little bit random because not every virus is actually viable and can be functional. But you know, we're looking at one E. coli cell and somewhere around the order of maybe 200 new viruses, depending on the type of virus, right? So why is this called lytic? So lytic, this word here, lytic, is kind of like related to the word lysis, and it means breaking. So the last step, I had mentioned that there's an enzyme in there that, that breaks down cell walls. And so this poor E. coli basically bursts open, and we've got hundreds of new viruses out there. So this is actually how a lot of diseases cause in humans. The virus goes in, it destroys your cells. Sometimes it's more of an inconvenience, other times it's life-threatening. So if you've ever had you know, if you've ever had let's say a sinus infection or influenza and you have a cough, those symptoms are from the virus kind of getting down into your throat and basically killing cells down there. And so you're coughing because you're irritated and, you know, from all the cell damage. If you have a cold sore, you're getting those blisters, and those blisters are caused from the virus killing cells. So that's the lytic cycle. Here's the video I wanted to show you. It's just really short here, but you can watch the process. It's kind of accelerated. I'll let it play for you. There's no volume. So here goes. There's E. coli just sitting there, all innocent. And here comes, oh, there it is. There's the bacteriophage. It's docking, and you're going to watch it inject its DNA. Kind of nasty if you think about it. There it goes. So the genetic instructions go in. More viruses are made. This takes, I think it takes an hour or two. I'm not exactly sure how long. I think it's 100 minutes. And then you can see in the background another infected cell is just exploding, and new infectious viruses are all over the place. So that is the lytic cycle, where it's lysing and breaking the cells open. I want to show you another strategy that some viruses use, and this is actually used by some of the herpes viruses. I mentioned they're lifelong infections, so sometimes they're hiding. So if you take a look at this, the very first step is basically the same. You have a virus, it's recognizing something on the surface of the cell that it's going to infect, and it's injecting its genome. So there's the little genome right there. And so in this case, something different happens. It can enter something called the lysogenic cycle. So I'll show you what that looks like. There's the viral genome. And the viral genome, you can see in this picture here, actually gets integrated entirely into the host genome. So this particular feature is something called a provirus. Sometimes it's called a prophage. So phage, bacteriophage, it's the name of a virus that infects bacteria, right? So sometimes people don't even call them viruses, they call them phage for short form. But provirus is the word to know. And then now it's a part of that cell. And the cell you can see here is replicating, and it's just growing, and that's it. The virus is part of that cell permanently. So this is why some viral infections are lifelong infections. For bacteriophage, there's different types, but eventually, often what happens is the virus eventually at some point does get its DNA, does get induced, which means suddenly we're getting viral RNA and viral proteins, and you can see this is entering a lytic cycle, and the virus is going to go and go infect someone else. So like I said, the human example is there's some herpes viruses that do this, and that allows them to have lifelong infections. You know, some people are probably thinking about HIV and AIDS, that's slightly different than this one, but similar principle. That's my next slide. So HIV is something called a retrovirus. So HIV stands for human immunodeficiency virus, by the way, and it does something really unique and interesting, which is why it is a lifelong infection as well. But if you take a look, it's an envelope virus, and you can see the envelope here is fusing with the host cell. It releases the capsid. The capsid actually dissolves when it hits the cytoplasm. And then we have viral RNA. So this is an RNA virus. And so this particular virus has an enzyme called reverse transcriptase. Reverse transcriptase. So that is what is going on right there. And so what does a reverse transcriptase do? It takes single-stranded RNA and it converts it to double-stranded DNA. So this is the opposite of transcription. Remember, transcription is where we take a DNA sequence and we make RNA out of it using RNA polymerase. This is the opposite, single-stranded RNA to double-stranded DNA. So, wow, this is weird. Why would it do this? Well, you know, we don't know. It works for HIV, so why not do it? Lots of different strategies out there, I guess. This is probably the main reason why it does it, is it does integrate into the host genome and becomes a provirus. So you can see there's our provirus there. So lifelong infection, unfortunately for people who have HIV, it is infecting immune cells and can totally, of course, cripple the immune system, which is what leads to AIDS if you don't get treatment for it. All right, so a couple things I want to say, just looking at the time here, about 5 minutes left, about viruses. And like I said, next day we're going to talk about COVID-19 and coronaviruses. But what about animal viruses in general? Because this is what we care about since we're animals, right? Well, there's no cell wall, so it actually makes it easier for cell or viruses to infect us in some ways. No cell wall to worry about. We also have a nucleus. Some viruses have to go to the nucleus. That makes it more challenging. And there seems to be a little bit more diversity when we're looking at animal viruses. Many different types of genomes. I think there are mostly DNA viruses with bacteria. So I want to show you, I have another little video I'll show you. It's 1 minute, and he's talking about animal virus life cycle. I'll play that for you right now once this loads up. All right, there we go. I'll play this.
Here's the virus particle with its envelope and proteins and genome. And you're going to see this represented as being within a pipette that's going to be dropped onto a petri dish containing human cells that are going to be susceptible to infection. Here's a blow-up of a single cell. Keep your eye here. These are the cell surface receptors. Here comes the virus particle that's going to bind. Here comes the disassembly stage. The genomes will, in this case, enter the nucleus. Not all viruses have to do that. And now you see replication. Here comes the assembly reaction. And these cells are now, the capsids are going to now go to the cell surface and bud. So what we've just witnessed is a single cycle of viral infection. A single particle infects a single cell and gives rise to many progeny. On average, 100 or so progeny per cell. Some viruses up to 1,000 or even more.
Okay, so I think there's a little picture here of an animal virus life cycle, and just want to point out a couple more things. It's very similar to what's going on with a bacteriophage. You've got that binding event, you've got a host genome getting in somehow. This one here, it looks like it's an RNA genome. And so the RNA, of course, is used as a template to make proteins, and some of these proteins are going to be made in the endoplasmic reticulum. So we talked about that, right? These membrane proteins are put there, so they have a signal sequence, and they end up right here on the cell surface. And then as the virus buds, it's going to take some membrane and those virus glycoproteins. And so this here is an envelope virus. So a couple more things just to finish off today. One is that, of course, a virus, I kind of alluded to this a few times, is viruses, they recognize something specific on cells. And so this means every virus out there has some sort of host or cell type of range. So for example, HIV is a very, very specific type of virus. It infects human cells only, and it infects something called CD4 T cells. That's not just any old white blood cell. That's a very specific type of immune cell. And so you can see there's an image there of HIV infecting that T cell there. So very, very specific kind of infectious target. Other things are a little bit more general. There's T4 bacteriophage, doesn't infect humans at all, but it infects E. coli quite well. Some have cross-species infectivity, so influenza virus can infect humans and birds and pigs. So that's why sometimes you might hear about avian flu. Avian means bird, or swine flu. Swine, of course, means pig. And, you know, so there's a risk that we can, you know, if you have a pet pig or live on a farm, you could infect your pigs, or the other way around, or we could have flocks of geese flying over us, you know, migrating in the winter, or sorry, in the spring or fall, and they could land here and infect us, or we could infect them. So that is true for influenza. Some viruses are a little bit more promiscuous. So rabies viruses seem to be able to infect you. If you're a mammal, rabies can infect you. Generally, it's carnivores because it's spread by bite. But there have been cases of rabid squirrels and things like that because it does infect just about anything. That's a mammal. Okay, so I'm going to just finish off here on one or two slides before I get into too much here. But I just wanted to show you the last thing here, and then we'll talk about, come back to this here next day. So I keep talking about this here, right? These processes. Like I said, they're very, very important. And so for viruses, they're also important for viruses, but it turns out because they have such a bunch of different weird genomes, they can kind of do things a little differently. So it says here they can be a little more creative with their genomes. So let's take a look at this. It turns out that for virus classification, there are 7 groups. So 7 different types of genomes. So some viruses have a double-stranded DNA, and this is probably not very confusing because it's just the normal process. We go from DNA to RNA to protein. Some viruses have single-stranded DNA, and you can see they need an extra step. They have to make double-stranded DNA first, and then our host cells will recognize it and make RNA and make proteins. Some have double-stranded RNA, and so that RNA can go right to the ribosomes. We can skip the DNA steps entirely. Some have, so we talked about sense strand and anti-sense strand and template strand. There's different strands in viruses. I'm not going to get into that, but you can see this one here is a plus sense RNA. It has to get converted to minus sense RNA, and then that can be used as messenger RNA. We also have minus sense RNA genomes. We have plus sense RNA genomes, and you can see this is that reverse transcriptase like HIV. So what is the point here? I know this is a lot of information. This is the point. Viruses have genetic instructions, and those genetic instructions are used to make messenger RNA, and that messenger RNA is read by host ribosomes in order to make more viruses. And that's kind of the bottom line. And there's a lot of ways to do it if you're a virus. So 450, I'm going to stop there. I have a few more things to say about viruses in general, and the next day we're going to talk about COVID-19, and vaccines. And I'm going to save a little bit of time for next day to talk about the final exam. So we are, I think, actually a week and a half away. If I've done my math correct, I think we're December 8th or something like that. So that's not too far. You may want to think about starting to study. And like I said, next Friday or next Wednesday will be all review. And if you have any questions before that time, please reach out to me. So have a great weekend. I will see everyone on Monday.